All right, everybody, we are going to start introducing today's webinar, continuing with this series during 2025, where we are highlighting and giving space to present the research done by our own very PhD students, which are working really hard, and they've been for the two and a half past years developing their thesis and their work. And today we have two speakers. We have Valeriia Ladyhina from the Swedish University's Agricultural Sciences and Adrián González López from Uppsala University, who are going to present their updates on their projects. And actually the first time that they will present to the bigger network and audience of the UAC, which we're very, very excited about. So today we will start with Valeriia. And before we do that, just a couple of housekeeping rules that we do always, which is this webinar is being recorded. So have that in mind in case you don't want your image, your camera, or your microphone to be recorded, then please write your questions in the chat and I will make sure to give them to the speakers during the Q&A section of their talks. But otherwise, you are more than welcome to just, put your hand up and then have a conversation with them, which I'm very sure that they will appreciate and like to have with you. But I will be here moderating in any case. More people are joining now. We will then change the screen sharing from my screen to you, Valeriia, to start presenting your work, which is the exploration of the resistome in pig farms here in Sweden and how this can contribute to the concept of One Health. And you just recently published your article with your methodology, which has been really cool. And we covered it in the last episode of the published podcast. And I'm looking forward to see all this kind of package in a presentation now. Thank you so much, Valeriia.
Thank you for the introduction. You see now my screen, yes?
Yes, we see your little friend there too.
Yes, so this is my favorite colleague among animals from one of the farms with whom we collaborate. And yeah, I am the only representative of SLU in our cohort, and I'm working with pigs. So I will talk today about the resistome in pig farms in Sweden as a contribution to One Health. But let's start with talk about antimicrobial resistance overall. So usually when we're talking about antimicrobial resistance to the average person, there will be maybe first three thoughts that are connected to the untreatable infections that are caused by resistant bacteria. The overuse and misuse of antibiotics is the main causing sign for appearance of the resistance in bacteria, and that we have the urgent need in development of new antibiotics. And what unites these three thoughts is that they're usually about human health. However, antimicrobial resistance is a bigger problem, and it involves many different players. Of course, humans, animals, and environment. But I want you to pay attention to one number. And it is this one. 70% of antibiotics that are used are actually used in animal sector. So animal sector is a very big player in antimicrobial resistance. So antimicrobial resistance is a One Health problem, which means that to be able to solve it, or at least come closer to controlling it, we need to approach it from three different perspectives. As I said, it's human, animals, and environment. And we also need to develop mechanisms and tools for active prevention, quick detection, and fast response to this problem. I am sure that many of you have seen these graphs where it is shown when antibiotics was developed, when it started to be used in human health, in human medicine, and when the resistance appears. But what is the history of usage of antibiotics in agriculture? So first antibiotics that was marketed for usage in animals was Prontosil in 1938 in Great Britain. And it is pretty close to the time when they were overall started to be used. Then in 1940, there was a mass outbreak of mastitis in US, and they treated it using gramicidin. Similar thing happened in Europe in 1943, and mastitis was treated with penicillin. And you can imagine that it was the wartime and there was like a very high importance of milk products. So they probably used quite a lot of antibiotics at that time. Then in 1948, the first antibiotics were licensed for usage in routine feed. And it was in US in poultry to prevent coccidosis. A bit later, antibiotics started to be used as growth promoters, also in US in 1949. And it didn't take super long to get the same strategy to Europe, just 1 year. So in 16 years, for example, in Germany, 80% of all mixed feed for livestock had antibiotics as a growth promoter, quite a big number. But luckily, in 20 years, in 1986, that was the first country that banned usage of antibiotics as growth promoters, and it was Sweden. The interesting thing is that among 10 farms with whom we collaborate, one of the farms was leading in opinion about the need of banning usage of antibiotics as growth promoters. So it is very interesting how now the strategy on usage of antibiotics and the resistance at the farm compares to other farms. And 20 years later, the ban covered the whole European Union. So here is the example on how much of antibiotics used in livestock overall in the world. And the example is from 2016. And you can see that there are some countries that have pretty high levels, such as Spain and Italy, some average, but Sweden, together with Scandinavia, have pretty low numbers. And that is the reason why our research is focusing on the Swedish livestock at the current moment. But what other factors that potentially contribute to development, persistence, and spread of resistance within the pig farm environment? Of course, we just talked that antibiotics is the main cause of appearance of resistance. But there are also other factors that are mentioned in the literature studies. And it is hygiene, temperature, and humidity. It also contact with wildlife as well as contact with pets. It can be feed because, for example, a lot of feed has additions of zinc or copper, which might potentially affect development of resistance or co-resistance. The quality of water resources, it is different visitors. For example, if some company that works with electricity visited 2 farms on the same day, they might transfer something on their equipment. It is size of the herd. It is organization of the box of the pigs. On this slide, you can see that the floor is straw covered. However, there is many different ways how the boxes can be organized. It can be concrete and it can be concrete covered with something, different materials from which the walls of the boxes made of, also size of the boxes, etc. And also organization of the farm in total. Is it the only one building in which all age of the pigs are located, and how do they, which rules they have when they going from one age of the pigs to the other, from one group to the other, or is it different farms that divide pigs of different age? So there are many different risk factors that might affect it. But before starting our research based on Swedish farms to understand which risk factors and how affect the resistance, we came to the question, which method is the best to capture the resistome, to identify these risk factors. Current methods that are used in research of antimicrobial resistance are culture and indicator bacteria or qPCR of selected resistant genes. Well, the first one have big limitation, which is that the 99% of bacteria are difficult to be cultivated or not culturable at all. The second one is still insufficient to obtain the comprehensive picture of the bacterial community and the resistome. And there is a very promising technique, which is metagenomics, which allows to describe total microbial community as well as identify known and novel antimicrobial resistant genes and mobile genetic elements. However, there is not so many studies yet based on metagenomics, especially if we are talking about non-clinical longitudinal studies. And this is connected to a few different reasons, to the complexity of the samples, the environmental samples, and the lack of standardized protocols on how perform sampling and further manipulation of the samples. So because of this, we decided that we need to look into methodology and think how we can optimize it for research on these complex samples for the purposes also for longitudinal studies. And there are several different critical steps in the pipeline of analysis of resistome, and they are next. So first one is sampling. The currently used samples are usually like swaps of individual pigs or picking up separate drop-ins, but then you come into the problem of randomization, as well as understanding how many samples you need to collect to get the description of the group rather than individual animal. And the more samples you collect, probably the higher cost for the analysis will be. Then the next question is transportation. Within our research, the current research within my project, we have farms that are located pretty close, but in order for the further research to get the good enough statistical power, we need to collect samples from probably like hundreds of farms. And then we need to know how it's better to transport samples that the transportation or temperature of it doesn't affect the taxonomic community, as well as resistome. Also, DNA extraction might fluctuate the outcome of the research, and the choice of sequencing technique. There are two techniques that we will compare in our studies. It is short reads and long reads. While short reads has small error rate, it is possible to have very deep sequencing with it, and it is cheaper than long read sequencing, if we're talking about the same depths, more or less. The length of the reads is still significantly smaller than the length of the resistant genes. But the long reads can help to solve this problem, while they have higher error rate and it is pricier. So what is the better option? And of course, bioinformatical analysis. There is plenty of different tools that is possible to use in metagenomics. So you need to know which one will be the more suitable for specific samples, for the complexity of the samples especially. And last but not least is the choice of the database for search of resistant genes. So the aim, as I said, was to develop a protocol for reproducible resistome research on complex metagenome samples from farm environments. And let's come to some results. So in our study, we suggest to use SOC samples as the novel method for AMR detection. You can see it on the figure, how it looks like, but I also have here one example. So it is like a piece of fabric that looks like a tube, and we put it on their boots. And after that, you walk within the space of the pig box, and by that collecting the sample. So SOC sampling before was considered to be sufficient for analysis of some pathogenic bacteria, but wasn't before evaluated for research of antimicrobial resistance. And that's what we did in our studies. So we think that SOC samples might be a good solution to remove the problem of randomization, because we more or less equally cover spatially the group of the pigs to get the whole picture of the resistome in this group. It's also easy and invasive method and cheap. Then I talked to you about the transportation. In our case, we compared 3 different temperatures: -80 mocking transporting of the SOCs on dry ice, +4 in cooling box, and room temperature mocking the transportation just by post. And here you can see that we collected samples from 3 different boxes of pigs, and then we compared the effect of temperature per box. So there is no visual effect of the temperature on the relative abundance of the resistant genes found within different classes of antibiotics. So what is the outcome of this part of the project is that the SOC samples give enough of material for analysis of resistance. However, you need to take into account that we did not compare SOC samples to individual samples, and short-term storage of SOC samples at room temperature does not have visible effect on resistome, which is pretty good. Then, comparison of DNA extraction kits. In our studies, we compared 3 different kits from Zimo, Omega Biotech, and Angin Biotech. And we compared them on 2 levels. First of all, we looked on the total taxonomy that we recovered from our samples with these 3 kits. And as you can see that 60% of the, I maybe need to switch on the laser. Yeah, so 60%, around 60% of total taxonomy that was recovered from our sample with all 3 kits was recovered by every single kit, while the kit from Omega Biotech recovered the biggest variety in taxonomy. The second step of comparison is the comparison of abundances of 40 most prevalent bacteria genera. And between different 3 kits, we don't see visual effect on that some of these kits missing some of their bacteria from this most prevalent genera. So based on this, we decided that the Omega Biotech is probably the best choice because it's recovering the biggest number of total taxonomy and because of this user friendliness. Next, sequencing technology. In our case, we compared Illumina sequencing platform based on 250 base pairs per end to Oxford Nanopore platform, which is called Me9. And again, the comparison of abundances of 40 most prevalent bacteria does not show that there is big effect of sequencing technique on what we recover. But Illumina overall recovered 30% more of different bacteria genera, which were more rare genera. And this might be connected to that the depth that we can reach with Illumina was significantly higher. In our case, it was at least 6-fold greater. So if you are focusing on the identifying the major part of the microbial community, deep sequencing with Illumina and sequencing with Me9, both are adequate for that. However, if the focus of your research is more rare species, you should choose deeper sequencing with Illumina. Then comparison of popular assemblers. Currently, you can use long-read assemblers, short-read assemblers, and hybrid assemblers. In our case, Cano and Metafly are long-read, Metaspate and MegaHit short-read, and HybridSpace and Opera MC are hybrid assemblers, which includes both long and short reads. For some reason, I was actually hoping very much for Opera that it will work good for our samples, but it totally didn't work on our type of samples. While the long-read assemblers also didn't show good results, and mostly we compared the quality of assemblies based on genome fraction, largest alignment, and total aligned lengths, because we want to save as much as possible data to lose as least as possible data for resistome analysis. And Metaspades, MegaHit, and HybridSpades, all of them gave us quite good result, with MegaHit having the longest total aligned lengths. We also compared the outcome of assembling based on search for resistant genes. And here it is again, HybridSpades, MegaHit, and Metaspades give us quite big variety of different resistant genes within different antibiotic classes, while Metafly, the long read, has quite small amount, and we also tested metaplasmid spades, but there we lose too much of information. So overall, we suggest that MegaHit performs the best together with HybridSpades. So if you have not a lot of funding, go for MegaHit. If you want to play a bit more around and you have funding for doing long reads, hybrid space will also work good. We also tried to add a step of binning after assembly in order, if we want later focus not just on the resistant genes, but also where these resistant genes are sitting. But unfortunately, adding this step results in loss of more than 50% of resistant genes that were found in assemblies. Next, very important question is how deeply we need to sequence our data, our samples. And for these purposes, we mocked sequencing at different depths, starting from 2 millions, ending with 19 millions per entries. And we decided that 85% is our cutoff of the total richness that we are aiming for, at least. So in order to get at least 85% of different gene families from total that were found in their sample, you need to sequence around 25 millions of reads. While if you want to look on the specific variants of antimicrobial resistant genes, you need to increase this number to 45 millions. And this data is fitting to the previous data that was obtained by Gvion, where they compared the sequencing depths of Illumina sequencing, but at the length of the reads, 150 base pairs. So this is good. And yeah, for families, 25 millions, for gene variants, 45 millions. Then, as I said, very important step is the choice of the database. In our case, we compared 5 different databases, Argonaut, CART, MegaRes, and Cibine and ResFinder. And among them, MegaRes gave us the best results. It resulted in the bigger number of unique sequences found in our samples, also of unique gene names. And it covered 84% of the whole resistome that was recovered with all these 5 databases. MegaHit and CART also managed to recover some genes to some antibiotic classes, such as aminocoumarine, for example, which were not found by other databases. And CBI was found more resistant genes within the classes such as aminoglycosides and glycopeptides and to others. And the additional plus of both CART and MegaRes is that with them you can also find a resistance to biocides, and MegaRes you can find the resistance to metals. And one minus that we found during our research is that the ResFinder database, for some reason, has quite some number of accession number duplicates. So if you work with ResFinder, you need to be sure that you didn't confuse by accession number the genes. But overall, the idea is that if you are focused on some specific group of the antibiotic classes, you might vary which database you want to use. But if you want to recover as much as possible to get as whole as possible picture of resistome, you need to use multiple databases in your research. So here is a suggested pipeline. SOC samples, usage of a megabyte kit, deep sequencing with Illumina, assembly with MegaHit, and search for resistant genes with the tool Abricate against several different databases. And now we will talk a bit more about not the pilot study, but overall the project. And the aim of my project is to explore the dynamics of antimicrobial resistance in livestock production as an important contributor to antimicrobial resistance in the environment and useful model for AMR studies. And to achieve this goal, we have 4 sub-goals. First, to describe the fluctuation and changes in resistome over time in the farms. Then to figure out how different treatment schemes with antibiotics affect the development of resistance. Also, look on the risk factors that I described before. How do they affect the development and spread of AMR within pig farms? And evaluate the significance of these factors. So here you can see the methods that are used in this study. As I said, it is the SOC sampling over 10 different pig farms within 2 provinces of Sweden. The sampling was ongoing monthly, starting from 1-2 weeks when piglets were just born, and then like 1-2 weeks before they were slaughtered. And then the samples are used for chemical analysis, environmental metagenomics, antibacterial activity to find out antibiotics and biocide residues and the phenotypic resistance of Escherichia coli and Enterococcus. After analysis of this data, we will compare it and combine it with epidemiological data on usage of antibiotics on the farms and other risk factors, and environmental parameters, temperature and humidity at the farm, and use advanced statistical modeling in order to figure out which factors do lead to the development of AMR in Swedish pig farms. And a bit of other results. This was done by a student whom I supervised on phenotypic resistance, where we collected isolates of Escherichia coli and Enterococcus faecium. On the left side, you can see the comparison of the resistance over the time among 10 different farms. And for Escherichia coli, you can see that there is a peak on the second sampling, which makes a lot of sense because based on what I have seen on the farms in Sweden, pigs are being treated mostly during the first 2 months. The first months when they were just born and they weak, and it's very sensitive period for them. And then the second sampling that was done here was a bit after they were weaned from their mothers. And this is also the period when a lot of them get in some infections, which is probably connected to that it is a very stressful period, plus they have change in feed. But after this, as you can see, for Escherichia coli, the pattern goes down, which is pretty nice. The usage of antibiotics in the first months quite quickly. The appearance of resistance due to the usage of antibiotics in the first 2 months quite quickly been reduced over the production cycle. While for Enterococcus faecium, we don't see similar picture. It's overall higher levels of the resistance, and it is a bit more stable over the whole production cycle. On the right part of the slide, you can see the comparison of the resistance over the whole production cycle per farm. And again, you can see that the Escherichia coli has visually a much less amount of resistant isolates in comparison with Enterococcus faecium. And we have even the farm where we didn't find resistance, which is very good. While for Enterococcus faecium, every farm had at least resistance to 2 antibiotics, and farm number 5 had even resistance to 5 of them, or to 4 of them, sorry. Yes, so there's quite interesting results, and I'm waiting to get the genotypic resistance results to compare it to see how they correlate between each other. And a bit about the progress. So what was already done in my project is, as I said, SOC sampling, and we already did the sequencing of environmental samples and also phenotypic resistance that was done by Linus. We also collected already all data on epidemiology and environmental parameters. And currently we are working on performing chemical analysis and bioinformatic analysis. So it will be very soon that I will be able to combine it all and start to make sense what's really happening in the resistome in Swedish farms and how different risk factors affect it. And this is the last slide. I want to thank to my project team, my 4 supervisors, Professor Susanna Stenberg-Leverin, Professor Johan Dixvedt, Elizabeth Rajala, and Professor Eric Bornkamp-Rudloff. And I also want to say a big thank you to Leila, who is a postdoc. She helped a lot during the pilot studies. I want to thank to SLO Future One Health for funding, as well to UAC for support overall, me as a PhD student of the project and promoting it at different platforms and conferences. And of course to you for your attention. And I hope that I managed to get your interest, to make you interested in this topic. So if you want to follow further research to know what genotypic resistance we have, how it correlates and which factors affect, please sign to the to my LinkedIn via QR code. So thank you for your attention.
Thank you so much, Valeria. You're getting some applauses at the chat. So great to see it all together, and now also put into the context of, what is it that you want to do with this method that you developed? Because you just have published, the methodology, but obviously you are going to apply that methodology to something, and things will get even more and more intricated as you're getting closer to your defense. If anybody has any questions, as I said, feel free to open up your camera and app. I think Divya might have a question. Uh, and Susanna is also here, which is one of Valeria's supervisor. Hi, Susanna. And let's have a discussion about this. Divya, go ahead.
I thought it was a great talk. It was nice to see you again. It's nice to see your results. Congratulations on your paper being published.
Thank you.
I had a number of questions, but you could stop me. I can email you also. But I had a few questions from the beginning. Like at one point, you mentioned after you do the binning, because in case you want to look at where the resistance genes are sitting, when you bin after assembling, you lose about 50% of the genes. I just wondered if like the 50% is across all the different assemblers, and also if this means that the genes that you lose are not actually found, or like what that implication is.
No, so the first question about after different assemblers, the search for the resistance after binning was already based on mega hit.
Mm-hmm.
We didn't perform it on other tools.
Mm-hmm.
And no, it doesn't mean that these genes do not exist there. It is the result of that at every step, like we lose some data.
Mm-hmm.
As you saw that like there is not all lens that we have initially sequence is aligned into assembly.
Mm-hmm.
And also when we do the binning, we also lose some data. So this is just the result. In the bins, we don't have quite a lot of data from the original amount of data.
Mm-hmm.
So
Yeah, so it's all still accurate. It's just the natural process.
yes, it's just like the more you process data, the more you lose in the end. I mean, like you can play around and agree which is good enough for your studies.
Yeah. But when we work with so many different tools, right, how do you validate that the results that you're getting are actually right? Like, for example, if you say that this database gives me more genes, how do you know that it's not like overestimating?
I think this is the same problem I have because like we can't really understand what is happening in our samples. But I mean, like still the major part of the genes that were found are like you can see that they are found in all databases. It is exactly the same sequences. But I compared also the sequences that I could retrieve from different databases to each other. And it's just that some databases have more of different varieties per one resistant gene.
Mm-hmm.
So I think that we need to assume that people who did their job on creating these databases and the tool did their job well.
Uh-huh. Yeah. So when you compare against the different databases, you look to see if like the results are consistent across all of them, then I suppose.
Partially, yes.
Mhm.
And as I said, the main part is consistent,
Mhm.
but there are differences. I mean, like just because the database has different information, and
Mhm.
as I said, like CART and MegaRes, they have additionally description of genes that are showing resistance to biocides and metals, which just do not exist in others.
Mhm.
So it's also possible like that some databases were historically focused on resistance to beta-lactams and they have more described resistant genes and their varieties in the database.
Oh, that was very interesting. Okay, last question. Just out of curiosity, when you talked about the second part about looking at like factors leading to AMR development, just out of curiosity, what statistical models are you thinking of using?
That's a very good question. but still a bit in future. So I didn't think that about.
That's okay.
I
Okay.
mean, it probably will be linear modeling, but
Mhm.
still something to discuss.
Yeah, I will look out for the next publication. No,
Thank
but
you.
congrats again. Thank you very much.
Or
Thank you.
Divya, you can just talk to her, like you
Yeah,
guys can
yeah.
talk while the things are going. So,
Yeah.
great. Susanna, did you have a question, or are you there also like kind of support and showing your face?
No, I'm just here to support, but I think these are good points that you're making, and we never know for sure, do we? We only know what we see, and I think you need to really try and look into why do I get these results? What could be behind them, and what are we missing, and what is still there, what's left to look at. And I mean, the only thing you know in research is that sooner or later you will be proven wrong. So you learn more and more. But I think we need to question our own results and sort of dig a little bit deeper into the tools that we are using. Because what Valeriia has done is actually look at exactly what she's done in every step. Because people are starting to use metagenomics on everything, and they use protocols that are already published without sort of questioning, are these the most suitable for my samples, and what is it I am looking at? What is it I want to find? So I think that's what we need to look at every time. Why am I doing this? What am I looking for?
Yeah, that was one of the questions that came up for me while I was listening. Now it's like, why are these databases biased, right? Like, what is the reason, like that some might show you these things versus the other things, or really understanding what is on the databases and why do you get the results that you get, then can give you a lot more information about your process as well. Really cool. Anybody else has any questions? Meanwhile, you guys either write or put your hands up. I did think about a couple of things. One question is about what, for you, Valeriia, what was the part that was more tricky, some or more difficult to optimize of all the different things you work with? Or maybe that it give you more headache, so to speak, to analyze and come up with a
I think
result.
I think the assembly part is the tricky one, but the only reason is that we work with pretty deep sequencing, and it just takes a long time to process. So it took a lot of time to process these 7 samples in all assemblers. And also, like some assemblers are very open to working with big amount of data. For example, MegaHit that we suggest can process at the same time data from several lanes, while Metaspades was crashing at that. So I needed to process sometimes it separately, a few times per sample. So even that's something that might affect how good is your research and how fast it is.
Mm-hmm. Mm-hmm. Yeah, that makes a lot of sense. A lot of thing that I was thinking from your process is, why would those kids that you look into recover different amounts of diversity? What is the difference between them?
I mean, like it can be how hard the bit beating is happening.
Mm-hmm.
For example, because the kids had the bits of different diameter, and
Mm-hmm.
in the protocols they described the different lengths of the bit beating. Just that step might affect. But also, like 2 of the kits were based on magnetic bits,
Mm-hmm.
while the Omega Bitech is based on the column filtration. And it's also like we also looked on how much data we are getting from the kit, how good quality it is, because it's, of course, like with all kits, I got enough of data to be able to sequence,
Mm-hmm.
but Omega Biotech also gave us the better quality, which means that it reduced the problems of precleaning of the DNA before using nanopore sequencing.
Mm-hmm.
So we didn't lose anything on that step. So that's also something might affect.
Mm-hmm, mm-hmm. Your overall result from the kids. Yeah, very interesting too. Sometimes we joke in science that, you get different results from lab to lab because the water is just different. So like imagine how many different steps of variability and possibilities of affecting the results are. So it's so cool that you have managed so many variables and looked into it and see what is the better supposed pipeline that people can use. And as Susanna was saying, always, but people have to think, is this really what I want to do for my samples in particular and for the data I'm looking for? Super cool, Valeriia, to see what you've been up to this past 2.5 years and coming into, the second strong half of the PhD. And I think with that, we're going to take a 5-minute short break.